Thank you for the invitation for this event. My name is Antti Kause. I'm the coordinator of the AQUAIMPACT. I will not describe what we do in AQUAIMPACT, but go directly to today's topic. I work in Finland, in Natural Resources Institute Finland, which is a governmental research institute. We do a lot of work with industries, and I have a background of, let's say, over 20 years on aquaculture breeding and genetics together with the industry. One peculiar thing is that in our research group, we also make software. We make the Mix 99 software, which is used for genomic breeding value evaluation, at least in 15 countries worldwide. And it means that we work on the breeding value evaluation data analysis quite a lot. So you know this as a background information. It's a commercial product. I start with the traditional sire dam offspring breeding. And this is the way that has been used for decades in the fish breeding programs. And it has been very successful. Of course, the logic is that we know either through DNA markers or family tags and ID tagging the sires and dams of the offspring. And we use this very basic information to select the best families and the best individuals within the families. And there has been a very good review about this, which you can, it's an easy reading review. I demonstrate some novelties in this area, what we have done in AQUAIMPACT. And the one important thing is the feed conversion ratio, which is typically very difficult to record. But we have shown that in the Finnish farms, in rainbow trout farms, the farm data, commercial data, shows that the feed conversion ratio is improved really considerably during the years. And with our breeding program data, which is really 8 generations of data during this time, the genetics has improved FCR also through selection of growth and body composition. And we are able to estimate that because we know how much genetics has improved and we know the phenotypic trend at the farms, that it's about 18% improvement in the FCR, which is actually quite a considerable change. And what it means is that the feed costs go down considerably, and also the total production costs. And it's easy to calculate that it's millions of euros, depending on the size of the production. And this has very positive effects on the environmental impact also. So there's less nutrition into the aquatic environment, like nitrogen and phosphorus. And in AQUAIMPACT, a similar study has been done, but by comparing the generation 0 fish to the generation 10 fish. And this is work done in France, in rainbow trout. And then it's just a comparison of 2 fish groups, in which we need to do less assumptions than in the Finnish study. But we actually see very similar improvement in FCR, and they have selected for better fillet yield. So there has been a genetic change also there. And then again, it's possible to calculate that there are millions of euros of benefits for the industry from this sort of selection. And all this is based on the traditional sire dam offspring pedigree. And today's topic is that, okay, how do we use genomics? And the basic conclusion is that with genomic information, we can do even better. And the basic approach to use genomics in breeding program is that we analyze the DNA of the fish, and we get DNA markers, what we call SNPs, which are the smallest particles of the DNA. And based on the thousands of these markers, we can replace the traditional pedigree with what we call the true genomic relationships between the animals. So it's not just like this sort of categorical sire, dam, and offspring, but really the true nature from the DNA, what the individuals have. And this is called a genomic selection or genomic evaluation. So we move from the pedigree-based data sets to something that uses the DNA profile or the genomic relationship of the individuals. And what does it benefit us? And an good example is the disease resistance, so that we have a biosecure broodstock in which we have fish, and we don't even want to have the diseases with these fish. So we are not able to record it. But the ships of these fish are in a, let's say, in a sea farm, and there we see the mortality due to a disease. And then we take the tissue samples and analyze the DNA. And then the DNA predicts the survival of the fish. And then the next step is that we genotype the broodstock fish, and we are able to predict the genetic superiority of the broodstock fish for the disease. And this means that with this method, genomic selection, we can breed novel traits like disease resistance and product quality traits or slaughter traits. And important is that we get more accurate identification of the genetically best fish. And if we get an accuracy increase of 10%, it directly means that we get 10% more genetic improvement in the trait that we select. So accuracy equals improvement in the genetic response. And this is a study on rainbow trout, and it's resistance against the Flavobacterium columnare disease, which is very common. And we made a big experiment in which we compared the accuracy of selection with the pedigree-based, the traditional way, and then the genomic evaluation. And we can show that we get 16% more accurate breeding values through this genomic information. This is a trait that needs genotyping anyway, so we anyway need to move with the methods that are something like this. This is at the lower end of the benefits what we see. If you want to see more examples, then the paper by Ross Houston and others is a very good summary of these. Okay, and then when we move to this genotyping, it increases the cost of the breeding program. And then there becomes an issue that perhaps we are not able to have so many fish in the breeding program. And for example, in our case, we have family tanks. And for us, it's very easy to ID tag the fish and make the traditional pedigree. And there is some sort of trade-off between how much we want to genotype and how much fish we want to have. And this is a formula which defines that how much genetic improvement we can have in traits. And there are two components in it, which are now important. One is the intensity of selection, so that if we have a lot of fish and we select only few of them, then we have very strong selection. If we have less fish, then the intensity goes down. So in case we move into a scheme where we have less fish and we genotype them all, then it doesn't necessarily bring the benefits what we want actually. And the genomic selection, especially in fish, it increases the accuracy. So if with the breeding, if with the genomic information we try to increase the accuracy of how we are able to define the genetic properties of the fish, then there may be a trade-off. And then we can ask that, can we somehow solve this? Or can we combine things? And it is very easy to combine both the traditional pedigree, which is still very useful, and then the genomic information, which comes from the genomic evaluation. So we can actually combine the traditional breeding and then the genomic evaluation into a single, let's say, computational effort. And it's called a single-step evaluation. And this is routinely used in animal breeding in livestock, cattle, pigs, chicken. And of course, the logic is that in both cases, we are talking about how animals are related to each other. And this is extremely useful. Another thing is that so far we haven't talked about regions or genes that impact traits. It has been like a black box approach, what I have described. But there's a peculiar thing that in fish, and in especially for disease resistance traits, we have genomic regions in the genome which have a very strong effect on the trait of interest. And we typically show them using this sort of what we call a Manhattan plot, where in certain chromosomes we see statistically significant associations with the traits. And these are very common for disease, surprisingly common for diseases in fish. And there is a long list of cases where this sort of single regions or SNP markers for different diseases explain a major part or significant part of the genetic variation. And now we have specific markers in this. There are very clear examples where single markers can have a big effect on the mortality. Then the question is that what is the consequence of this? And there are at least two consequences. One is that if we are able to really validate in several data sets in our breeding program that these markers really function always in the same way or more or less same way, and there are many of these markers that determine the traits, then we can do something called SNP picking. So we just use a few number of these markers in the breeding program, and we reduce the genotyping costs. There are some examples of this. This is not an easy task and not very probable scenario in many cases. Another thing is that with the genomic evaluations that I previously described, we assume that this doesn't actually happen. And then it creates a problem. But it can be again solved. So we had the pedigree, the genomic relationships, and now we can add on the top information about this, what we call QTLs, so specific markers. And that is called single step QTL weighted genomic evaluation. And this again describes the power that we can combine things. And there are a lot of different methods to do this. I want to stress this because this is the most potential way to analyze multiple traits simultaneously. And we actually tried this approach. This is the same data as in the previous slides. We just now add on the top of this that we specifically give emphases to the regions that are linked to the mortality against this disease. So they get more emphasis in the breeding value evaluation. And we get a small increase, not much, but small increase when we fully utilize the information. There are cases to do the same, and in some cases the increase, let's say from the pedigree-based to the weighted genomic evaluation, it's much bigger. But and this depends, of course, on the size of these effects for single SNPs. If they are small and they don't exist, this doesn't help. If there are large ones, then it will help more. If there are less markers, then perhaps this works better, and so on. So it's specific to traits. Okay, then the second last slide. So I described that there are multiple ways of using genomics in the breeding programs, and they are routinely used, both in aquaculture and in livestock especially. The question currently is that not in a way that what is the best way, but for a breeding program, the question is how to optimally use these. And we don't always need to take the most sophisticated way. And now linked to this AQUA-FAANG. So what I have described, we have not needed to use information about the genes themselves, their functions or gene expression or gene interactions or epistasis. It's a in a way blind way to use the DNA. And then the question is that can we do better, or can we generate new tools? And this is where the AQUA-FAANG comes in. That it is possible to make new things to improve these described methods. And I will not list here things, but just to show that there are avenues, and AQUA-FAANG people are generating stuff that can be helpful. Thank you.